All right. So as you noted, my name is Jay. I'm from the Williams Lab here at Georgia Tech, and so I'm going to be talking about ribosomal evolution today. Okay. So in the Williams Lab, we like to do research on translation in the ribosome because these processes are universal. All life depends on coded protein in order to do its life cycle, and so the process for creating coded protein is translation, where we go from RNA to our protein that does the work in the cell. And at the center of translation is the ribosome. So it's like the nexus that collects everything together that gets translation done. So it's the focal point of this incredibly important life process. And so not only is translation in the ribosome universal, it's also central and necessary for life. And so what this means in terms of evolution of the ribosome is that it's a process that began at the very root of the origin of life. So it's older than life as we know it. And so using this insight, our lab has created a model for the evolution of the ribosome called the accretion model. So that's this figure you see on the right of this slide right here. So as I mentioned earlier, this is a process that started before extant biology. So, the beginnings of it was billions of years ago. And this is a process that if we continue up this kind of model timeline, at the very end here, this is still an ongoing process. So it's a process that spans multi-billion years. But for the purposes of today, I'm just going to be talking about a smaller portion of that multi-billion years that focuses on ribosomal structure. And so the basic idea of the accretion model is that evolutionary process is based on this idea of building on top of things that came before. And so specifically what I'll be talking about today is the early ribosome growth and evolution that happens through accretion of structural elements. And so whenever I'm talking about structural elements, specifically what I'm referencing is, what I'll be referencing is RNA structural elements, and I'll be calling those RNA structural elements specifically ancestral expansion segments. And so, as I said, the idea is that accretion is this process by which we don't perturb structure that came before, and we build upon and elaborate more structure on top of that. So I just kind of briefly introduced our model for ribosomal evolution. So my goal today will be to characterize what is the first 5 AESs or ancestral expansion segments within the accretion model. And so in terms of experimental system, that translates into 6 RNAs that are some subset of the first 5 ancestral expansion segments, which I've listed those RNAs here. And so if we look at this figure on the right, we can see the 3-dimensional structure of these ancestral expansion segments as they appear in the ribosome. And so, for example, in Figure A, we see the accretion event whereby AES2 accretes onto AES1. And so we can see in red this helical structure here is AES1. And then we have the accretion of AES2 on AES1. And as I mentioned earlier, a feature of this process is that the underlying pre-existing structure, in this case AES1, is unperturbed, even though we've kind of added on this extra structure. And then we can see in other panels accretion of AES3 on the AES1, AES4 accretes onto AES3, AES5 on 3. And then in this panel D, we see secondary structure, which this is just a 2-dimensional representation of structure of RNA that we'll sometimes be using in lieu of the 3-dimensional structure. And then here in the bottom, we have a 3-dimensional representation of all 5 of the ancestral expansion segments together. And so, so in terms of the methods that I'll be talking about today, I'll be talking about UV melting experiments. And so the basic idea of that is we're going to monitor the absorbance of our RNA signal as we change the temperature, and that'll give us a melting curve. And then we'll take the derivative of that melting curve and we'll fit it to a model that'll let us see the transitions occurring within that RNA, and we'll use that as like a proxy for viewing the structure within that RNA. And then we'll also look at SHAPE, which is a structure probing method, which I'll go into more detail later. So first I want to talk about melting. So I'll start off by saying that these experiments and the figures you see on the slide were done by a previous student in the lab, Catherine Lanier. And so, so maybe first we'll start off by looking at panel A here, which this is, we see the structure of AES1 in this panel, and then we also see in this kind of solid colored line, we see the derivative of the melting curve of AES1. And then in the dotted line, we see the fit of that curve, and then in the solid black lines, we see what we're really interested in, which is the individual transitions that make up that fit. And so these are basically, we're basically looking at, you know, if we consider of this profile, this melting profile, it looks like we have two major transitions. We suggest that these two transitions correspond to the two kind of helical regions within AES1. So as I mentioned earlier, we're kind of looking at these transitions as a proxy for the structure in these RNA elements. And so our prediction based on the accretion model is that because we're adding structure on top of structure, we should see new transitions appearing with these accretion events. And so, for example, if we consider the AES1 profile, and then we look at AES1, 3, which is the accretion of 3 onto 1, we see indeed that there is an extra transition that appears. So here we have like 3 major transitions. AES1, we only have 2. So I'll go ahead and say that we see the same trend. So the trend we saw with AES3, we see that with the accretion of AES4 as well down here. However, we don't really see this trend whenever we accrete AES2 and then down here AES5. And essentially what we think this boils down to is the fact that AES2 and 5 are smaller structural elements. They're only about 20 nucleotides long. And so essentially what that means is that those structures are small. And so whenever we melt them out, we expect to have a very sharp transition, a very sharp kind of peak. And so it's likely that those peaks are being covered up by these larger, broader peaks that are associated with larger structural elements. So that's one part of the story. And the other part of the story that our model predicts is that we shouldn't, that the previous existing transition shouldn't be perturbed at all. So that's the prediction. However, the picture that the data shows is much more complicated. So once again, if we focus on AES1, we see we have 2 kind of major transitions here, one at around 70 C and one at around 60 C. And then we see, if we once again compare with AES1, 3, there's at least one transition that looks like it's been shifted. And we see this trend in all of the melting profiles where there's a peak that's shifted in terms of its temperature range, or the peak is broadened. So some kind of changes to the transitions happening. And so on the one hand, this seems to suggest that maybe there's a disagreement with our model. But however, if we consider the structure of the ribosome that corresponds to AESs 1 through 5, and we map on the base pairing interactions of that region of the ribosome, as we see in the magenta lines here in this figure on the left, what we see is that this region of the ribosome is actually really, these nucleotides are really integrated in terms of their base pairing interactions. And so while the overall structure may not be perturbed whenever we accrete a structural element, we're still kind of changing the overall molecule in the sense that we're adding base pairing interactions. And so what that ends up doing is changing the thermodynamic properties of these structures. And so while the 3-dimensional conformation might not be changing, we are perturbing the transitions to those thermodynamic properties. And so that's why we're seeing shifting and broadening here. So we think that still agrees with our model. Okay, so the last thing I want to talk about is shape. And so the basic idea of a shape experiment is that you have structured RNA, you add a reagent, and specifically this reagent will selectively modify nucleotides that are not base paired. And then we do reverse transcription reaction, and basically what ends up happening is wherever there's a modification, the RT likes to stop, and that creates fragments. And then we can analyze those fragments to get an idea of which nucleotides within our RNA tend to be modified and therefore tend to be not base pairing. And so, okay, so this is like the broad kind of global perspective of the shape data I've collected. And there's a lot here, and so for sake of brevity and clarity, I'm gonna kind of just focus on one region to kind of illustrate comparisons. And so on the left, we see the shape profile of AES 1 through 5. So this may look like a jumble of circles and colors, but the idea is that this is the secondary structure of AES 1 through 5, and essentially what we've done is we've made each nucleotide a little circle, and each circle has a color associated with it, and that color denotes a shape reactivity. So it's telling us what's the likelihood of that nucleotide for forming base pairing interactions. And so we see like really dark colored, dark blue, and that's like low to no shape reactivity. And then as we move to warmer colors, like you can see a light blue there to like a green to like a yellow, we're increasing the reactivity of our nucleotide. And so I've circled this region of the structure, and this is the region of interest that I want to focus on so that it's a little easier to see patterns. And so I've kind of reproduced this region for each of the 6 RNAs that are in my kind of experimental set over here to the right. And basically we see a trend whereby the nucleotides in the region around position 80 tend to have heightened reactivity compared to nucleotides in the surrounding region. And so we take this as indication that, you know, our experimental set is consistent in terms of its structure, in terms of its base pairing, but also what we're interested in is whether or not there's agreement between this experimental system and the structure of the ribosome itself. And so if we look at the corresponding region of the ribosome, the region that corresponds to what we're looking to over here, we see that, you know, once again, base pairing interactions with these magenta lines, we see that the structure of the ribosome agrees and that the region that corresponds to this position 80 over here is not forming any kind of base pairing interactions. So we see not only does our experimental system agree with itself, but it also looks to agree with the base pairing as we expect it from the structure of the ribosome. Okay. So with that, I want to conclude and just go ahead and say broadly, we think that the data that we're collecting agrees with our model for accretion, evolution by accretion for the ribosome. And it seems like we've been able to resurrect some really truly ancient RNA fragments. And even more broadly, we've shown that we can use some biophysical approaches to help us answer some really deep evolutionary questions. And so with that, I will say thanks for listening. I'd like to give credit to, of course, my advisor, Lauren Williams, for helping me out with this project. Anton Petrov, who's a research scientist in our group, did a lot of like the foundational work that led to this work. And then, as I mentioned earlier, Catherine Lear also made contributions. And then I'd also like to recognize support from NASA in the form of the Center for Chemical Evolution down here, and then the Center for the Origin of Life. And I'd be happy to take any questions.
Great. Do we have any questions?
All right. So even just in this sort of limited thing, you've got this limited region, you've got a couple of segments, and presumably there's and so even with just 5, which I think was the number you had, you start getting crazy combinatorics about which ones you could put together. Part 1, is there a reason for choosing exactly which sets that you chose? And part 2, is it possible that by looking at which additions produce the smallest changes when they're added, might that have relevance to which additions actually occurred in what order in actual history?
That's a great question. So I kind of wish I had time to talk about this. I cut out some of the background just for the sake of time, but all the details surrounding that is kind of built into the model. But the basic idea is that, the reason we were approaching this accretion idea of evolution of the ribosome is because if you look at extant ribosomes, they're not all the same. So like human ribosomes and E. coli ribosomes, our ribosomes are actually larger in terms of their ribosomal RNA. There's an increase in size. And so basically what people have seen is that, even if you look at extant ribosomes, we can kind of see this process whereby, new regions are added onto old regions. And you can actually tell that certain regions came before others because as we look at this, this slide, because of the way the interactions are within the ribosome, there are certain structures that had to exist before others. So like the metaphor is to think of it like, if you're looking at a tree, it's very obvious that the branch off the trunk clearly grew out of the trunk, and it had to have existed after the trunk. And so we kind of use that mentality of this kind of dependency of structure, and that's the whole basis of the numbering of these ancestral expansion segments. So it's all structure based, and the ordering of the combinations is very dependent on that process.
Great, thank you. So we have to move on, so let's thank our speaker.